This is the first of two lectures relevant to lipid metabolism. This one is lipid oxidation, or to be more specific, beta oxidation. The fatty acid has a challenge, that is, it is found in the liver cells as well as in the adipocyte. That's where it is stored. While in terms of its being utilized, it needs to be mobilized from liver as well as adipocyte and brought to the muscle cells. In terms of how is it mobilized, it needs to be in the form that is it's free fatty acid. That means it's not esterified. And if it is free fatty acid, it can be either mobilized by the use of albumin. Albumin is the major protein found in the blood, the most abundant protein in the blood. And this albumin can connect with the free fatty acid, the long chain fatty acid, and then mobilize to the muscle. While the short fatty acids, since they are amphiphadic, therefore they can just dissolve in the blood and be brought to muscles. In terms of the esterified type of free fatty acid or esterified fatty acid, which can be forming ester bond with to become triglyceride, also in terms of link up with cholesterol to be cholesterol ester. So these are a bit more complex and they are not easily dissolved in water environments such as blood. Therefore, they need to be brought to muscle by using the specialized transporter, that is lipoprotein. So once the fatty acid already arrive to the muscle cell, myocyte, it needs to be activated. Activation is to be connected to connect the free fatty acid to the coenzyme A. Therefore, it will be now known as acyl-CoA. And this reaction is done by the enzyme acyl-CoA synthetase, and that is located strategically outside of the mitochondria. The location of beta oxidation is within mitochondria. Interestingly, after it becomes acyl-CoA, in order to go from cytoplasma to inside the mitochondria, it needs to use this shuttle, the carnitine shuttle. In order to do that, it is removed from the coenzyme A and then attached to the carnitine. This carnitine is required in terms of moving the acyl or the fatty acid component from the cytoplasma to within the inside of the mitochondria. So once the acyl-carnitine goes through the translocase, then once it's inside the mitochondria, the removal of carnitine takes place and reattachment of the coenzyme A takes place again, and therefore you get the acyl-CoA already inside of the mitochondria to progress to the beta oxidation. So what I have explained in the slide previously, slide before this, been explained in text for you to read and understand this slide as well as this one. So now we look into the beta oxidation itself, where there is 4 steps. This beta oxidation basically takes the fatty acid. The fatty acid has already been activated now. That is fatty or acyl-CoA, such as palmityl-CoA, where it can be cleaved 2 carbon at a time, where 2 carbon at a time remove as acetyl coenzyme A. And in order to each time to remove these 2 carbon, it has to undergo 4 step involving the reaction at the carbon 2 and 3. Therefore, that's why it's known as beta oxidation. And if fatty acid such as palmitate or palmityl-CoA undergoes beta oxidation in series, you will have 7 beta oxidation steps, and it will lead to 8 acetyl coenzyme A from palmitic acid. So here shown the beta oxidation, whereby there are 4 steps for each time to remove 2 carbon as acetyl coenzyme A. So you have the acyl coenzyme A, where the reaction takes place at the alpha carbon as well as beta carbon. Therefore, the name beta oxidation comes about. It starts off with oxidation of this acyl-CoA, and since it involves a dehydrogenase, it's a redox reaction where oxidation is also accompanied with reduction. So in the reduction, you will have FAD becoming FADH2. And then the next step is hydration, where the trans-enoyl-CoA will undergo hydration, and it's followed immediately of another redox reaction where it will undergo oxidation as well as reduction, the formation of NADH, and the ketoacyl-CoA will then ready to be cleaved and remove 2 carbon to become acetyl coenzyme A, and the acyl CoA now has less 2 carbon, and it doesn't stop at this. It will keep on going another 4 steps and remove 2 carbon, and this goes on until all the carbon is converted to acetyl coenzyme A. So if you have palmitic CoA going through this beta oxidation, the total number of what you call cleavage is 7, while the yield is 8 acetyl coenzyme A in total. Not only that, since it undergoes 7 cleavage or 7 of these 4 steps, therefore you should be able to have 7 FADH2 and 7 NADH. So here given more description of that 4th step itself in terms of the mechanism. In terms of the acyl-CoA, it needs to be at each time undergoing beta oxidation to remove acetyl-CoA. But before it goes to the next cleavage, the first cleavage will undergo the 4th step first. And the first step is the oxidation and followed by reduction. Next one is hydration. All this happen at the carbon 2 and 3. This is then followed by another reduction and oxidation, and then followed by the cleavage itself to remove the acetyl coenzyme A. In terms of the enzymes relevant, this is the dehydrogenase. You also have the hydratase, dehydrogenase acyltransferase, and cofactor or substrate that is involving this 4th step includes FAD becoming FADH2. You also need to use the substrate water for hydration, NAD plus, as well as the last step using coenzyme A. So apart from me describing to you in detail by using the diagram, again here it's been given in text so you could read and understand in the few slides that's been given here. So we already covered in terms of short chain fatty acid, how it will undergo beta oxidation, such as palmitic acid entering beta oxidation to give rise to 8-acetyl coenzyme A. But what about other type of fatty acid? This includes odd number of carbon, such as 17 carbon fatty acid, 19 carbon fatty acid, and then also what about the fatty acid which is slightly longer, and also in terms of fatty acid that has double bond and without double bond. So we have to also look into the what happens to them before they can undergo beta oxidation. For this slide, it's describing in terms of the odd number of carbon fatty acid, such as 17 carbon, where this 17 carbon fatty acid will undergo beta oxidation as described just now. It will come to the last 3 carbon and it will be not going through any more beta oxidation, but instead the propionyl CoA will be converted by a series of reactions to become succinyl CoA. No need for you to remember the steps because it's quite complex, but end of the day the propionyl CoA will be converted to succinyl CoA, and succinyl CoA is the constituent of citric acid cycle, therefore it already enter the catabolic pathway. The statement at the bottom of this slide is referring to propionyl CoA can be entering the gluconeogenesis pathway to make into glucose. So from fatty acid, you can convert into glucose. So here shown the fate of the propionyl CoA that's been obtained through the beta oxidation of odd chain fatty acid, such as the 17 carbon fatty acid, where it will end up as propionyl CoA, and through a series of complex reaction it will be converted to succinyl CoA and therefore enter the citric acid cycle, and if required can also make glucose through gluconeogenesis. So now let's do the calculation of the total number of ATP can be obtained if palmitic acid undergoes beta oxidation followed by electron transport chain. You already know that palmitic acid will undergo 7 of the 4 steps. Therefore, there will be 8 acetyl coenzyme as well as 7 FADH2 and 7 NADH. You know also that each FADH2 undergoing electron transport chain will yield 2 ATP, while NADH will yield 3 ATPs. So if you put together the 7 FADH2 multiply by 2, you get 14, and then 7 multiply by 3, you will get 21. So 14 plus 21, you get 35 ATPs in total for the 7 cleavage or cut that is happening on the palmitic acid. Next, we look at the fate of the acetyl coenzyme A. So now that you have 8 moles of acetyl coenzyme A, each undergoing citric acid cycle will lead to 12 ATPs. Remember, citric acid cycle will give rise to, for each acetyl coenzyme A, 3 NADH, as well as one FADH2 and also one GTP, which equals to ATP. When you put together these numbers into electron transport chain, it will be 12 ATPs in total. So now you have 8 acetyl coenzyme A, which goes into the citric cycle. What you get is 8 multiplied by 12, therefore it is a total of 96 ATPs. So once you have these 96 ATPs, then you add with the 35 ATP, that will be 131 ATPs in total. But as you know, there need to be activation of the free fatty acid, and activation is adding coenzyme A. And in terms of palmitic acid, when you add one coenzyme A, you will lose 2 ATP. So from that 131, you minus off this 2 ATP for the activation of the palmitic acid. What you have is total of 129 ATPs net. These once been calculated is only 68% of energy that you get from the oxidation of palmitic acid. It doesn't give you 100%, and the remainder of 32% is in fact lost as heat. In this slide, interestingly, I will prove to you that fatty acid, oxidation of fatty acid actually give rise to twice as more energy compared to the oxidation of carbohydrate in terms of oxidation of glucose. So we will use the hexanoic acid, fatty acid with 6 carbon, and compare it with glucose, which is also with 6 carbon. So hexanoic acid will be firstly activated, and therefore compared to palmitic acid, here you lose only 1 ATP in the activation of hexanoic acid, since it is shorter than palmitic acid. Next, you need to cut or the cleavage of hexanyl CoA using that 4-step, and this only need to be cut 2 times rather than 7 times for palmitic acid. So once it's cut twice, it will lead to 3 acetyl coenzyme A, and 3 multiplied by 12, you will get 36 ATPs once it goes into the citric acid cycle. You also have for each cut 1 FADH2, since total of 2 cuts, therefore 2 FADH2, and this will lead to 4 ATP. Similarly, 2 NADH will lead to 6 ATP once it goes through the electron transport chain. Then we add together all the ATPs, you will get 45 ATPs in total in the oxidation of hexanoic acid. In terms of glucose, you're already well aware. You've been taught about it in the lecture for glycolytic pathway, as well as citric acid cycle and electron transport chain, where you will get a total of 38 ATP, but at times it could be also 36 ATPs. But it's still low compared to the oxidation of hexanoic acid. So how is it twice as much as energy being released for the oxidation of hexanoic acid in comparison to glucose? We have to use the molecular weight, the relative molecular weight, where 1 mole of hexanoic acid has 116 gram, while glucose has 180 grams. So you take these 45 ATPs, divide by 116, you should able to get 0.4. Do the same, 38 divided by 180, and you will get 0.22. So you can see that this is in fact the zero point 0.4 is in fact twice as more than 0.22, therefore proving that in every gram of hexonic acid being oxidized, you can actually achieve double the energy in comparison to the oxidation of glucose. The reason behind it is because glucose is in there's more oxygen, while exonic acid is in fact much more in reduced form than glucose. Therefore, giving more chances for oxidation of exonic acid to yield more energy. And next, we move on in terms of oxidation of long chain fatty acid. It's not really that long. It's actually 20, 22. It's not like 40, 50 or 60 number of carbons. So in terms of long chain fatty acid, it needs the help of another organelle, that is peroxisome, rather than to be done in the mitochondria. So to look at that, we have to move to the next slide. Here you can see the oxidation of long chain fatty acid will involve the peroxisome, or also known as glycosome. Here only shown the half of the peroxisome as well as half of the mitochondria to make a comparison. So the issue is in the long chain oxidation, say arachidonic acid with 22 carbon, it will have some losses as well as some challenges, where the loss is in the loss of FADH2, because the peroxisome oxidation of long chain fatty acid will actually yield hydrogen peroxide. This hydrogen peroxide is quite toxic, and by the use of catalase as well as the use of the reduction powers of FADH2, it will be converted to something safe, which is water and oxygen. And then you also have an issue where the NADH being achieved in the peroxisome as well as acetyl coenzyme A cannot be utilized further in the peroxisome, and therefore need to be brought out and then exported back to the back to mitochondria, where it can then be utilized. Here we look at the oxidation of unsaturated fatty acid. The unsaturated fatty acid, such as oleal or oleic acid, require to undergo beta oxidation up to the stage where the location of the double bond. So you can see that 3 beta oxidation steps will be done before it reaches the double bond at the location of carbon 9. And then what happens is, with the presence of another enzyme, that is enoyl-CoA isomerase, this enzyme will do some changes to the dodosinoyl-CoA, where it will then convert it to something of the intermediate of beta oxidation, and hence it will then enter the beta oxidation followed by this conversion. Here the oxidation of multiple double bond or fatty acid which has more than a single double bond is being described. In terms of example is linoleic acid, which has 2 double bond at location 9 as well as location 12. Again, beta oxidation will start off. 3 beta oxidation cycle will be done, and then once it's reached the double bond, the enzyme enoyl-CoA isomerase will then change it to something of an intermediate of beta oxidation, and therefore it is now will be part of the beta oxidation. Then you have also presence of additional another enzyme that is 2,4-dienoyl-CoA reductase doing some changes to the intermediate by using the reduction powers of NADPH. This NADPH we get from pentose phosphate pathway. So once the intermediate has been modified, yet again, enoyl-CoA isomerase comes in and makes the change to be part of beta oxidation intermediate, and therefore it then can continue into beta oxidation without any more issues, where there is no presence of any double bond to stop the beta oxidation. Here I talk about the regulation of lipid metabolism. To get direct to the point, it involves the synthesis on the left that is happening in the cytoplasma, while on the right, in terms of inside the mitochondria, the beta oxidation itself. So two different situation where one is high blood glucose right after diet. That means the person taking up a high carbohydrate diet, where it will be converted to glucose, and glucose in turn will be made into acetyl coenzyme A by glycolysis. When there is lots of glucose molecule, pancreas will release insulin, and this insulin will travel from pancreas to muscle cells, and it will activate phosphatase. Once the phosphatase is activated, it will remove the phosphate from this important enzyme for fatty acid synthesis, that is acetyl-CoA carboxylase. Once the phosphate is removed from ACC, it will become active. When this enzyme is active, it will convert all the acetyl coenzyme A to become malonyl CoA. So the concentration of malonyl CoA eventually will increase, and it in turn will cause inhibition of a crucial enzyme in the transportation of fatty acid from outside of the cytoplasma to the inside of the mitochondria, whereby it will disturb the carnitine shuttle. So the elevation of malonyl CoA will inhibit the carnitine from bringing the fatty acid from the mitochondria, from the cytoplasma to the mitochondria, therefore disallowing beta oxidation from happening. The malonyl CoA will be then converted to fatty acid, and fatty acid will be changed into triglyceride and stored. So that's fatty acid synthesis because of excess of glucose in the blood. So now we look into what happens if there is low blood glucose. In terms of starvation, fasting, there will be low blood glucose. Therefore, the body needs to resort in using fatty acid for energy. When there is low blood glucose, pancreas now will secrete glucagon. The glucagon will then activate protein kinase A. The protein kinase A is an enzyme that adds phosphate. Here it adds phosphate to ACC. ACC now become deactivated. Therefore, since in the initial stage there's so little glucose, therefore through glycolysis there will be less acetyl-CoA to be formed, and in turn, since the ACC is deactivated, less malonyl CoA will be made. Since there is so little malonyl CoA, it cannot go and inhibit the carnitine shuttle, and therefore fatty acid can now enter into the mitochondria, and the fatty acid can be then undergoing beta oxidation to provide lots of FeDH2, NADH, as well as importantly acetyl-CoA for citric acid cycle to take place. So at the end of it, because of the effect of low blood glucose, what happens is the cell will utilize fatty acid for energy rather than using glucose. Here you have another oxidation of fatty acid that is referred as alpha oxidation. I'm not sure whether I'm pronouncing it correct, since it's not really a symbol of alpha altogether. It has a little bit of difference in terms of the symbol of alpha, but basically we will pronounce it as alpha oxidation. In terms of alpha oxidation, it looks into fatty acid that is slightly modified, that is pythanic acid. This pythanic acid, unfortunately, since it has the beta carbon with a methyl group, therefore it cannot be entering the beta oxidation. Firstly, it will undergo alpha oxidation, where the modification will take place to make it remove that methyl group from the beta carbon. To do so, it will cause the activation of acyl-CoA synthase, where it will add coenzyme to pythanic acid to become pythinyl-CoA, and then further few steps where at the end of it there will be a cut happening to remove one carbon. When that happens, the methyl group no longer in the beta location, and therefore since the beta location is now free from the methyl group, and it means it can then enter the beta oxidation. So modification is done by alpha oxidation before the pythanic acid can undergo beta oxidation.